Chapters 5 and 6 have topics that kind of go together. In Chapter 5 will concern the topic of gene mapping, and Chapter 6 concerns the topic of genetic testing or genetic diagnosis. In Chapter 5, gene mapping chapter, we will be determining where a gene is located, and we'll be learning how to measure the distance between genes. Gene mapping is also sometimes known as a linkage analysis, and the most important aspect in Chapter 5 will be learning how to determine the distance between two different genes to determine whether two genes are linked or not. That is, how close together or how far apart are two different genes, and will they travel together during inheritance. That is the main topic in Chapter 5. Now, near the bottom of the slide, where it indicates the importance, this is the reason why we do such gene mapping or linkage analysis studies. One thing that gene mapping information helps us to do is to find the location of disease-causing genes that can help us to clone those genes, for example, for their study. And another very important development associated with the gene mapping information is a development of genetic diagnostic tests, genetic testing for genetic diseases. Now, in Chapter 5, we will spend most of our time talking about how we measure the distance between two genes, and then in Chapter 6, we will mostly talk about how we apply that information for genetic testing. Some of the other topics, such as using this information for cloning the genes, will be talked about in the biochemistry section of the class. Now, one of the first topics that comes up is what kinds of genes do we map? When we do gene mapping or linkage analysis studies, what kinds of genes do we study? Your first reaction might be that, of course, we study disease-causing genes because those are the most important to us. Well, it is correct that disease-causing genes are very important to us, and we want to include them in such gene mapping studies, but disease-causing genes are not always easy to work with. And so, most of the time, gene mapping studies are done using special kinds of genes that are known as DNA markers, DNA marker genes. And this diagram illustrates the four major kinds of such DNA markers that are used in gene mapping studies. These DNA markers are special kinds of genes that don't necessarily have any function at all. They don't necessarily do anything or code for any protein. The importance of such DNA marker genes is that we can find them in the laboratory. We can identify them in the laboratory by such tests as Southern blotting and PCR and so forth. And the other important thing about them is that they are highly polymorphic in the population. That is, for each of these DNA marker genes, there will be at least two different alleles, two different DNA sequences for that gene in the population, and we can detect those differences, those different alleles, by our testing methods, Southern blotting and PCR, for example, to follow inheritance of these alleles from parents to children. So that is the reason we look at these DNA markers in gene mapping studies. Well, what kinds of DNA markers are there that we study? Panel A describes a kind of DNA marker gene known as an RFLP, or restriction fragment length polymorphism. This means that in the population, for such an RFLP, restriction fragment length polymorphism gene, there will be at least two different DNA sequences for that gene, two different alleles for that gene that can be cut differently by restriction enzymes such as EcoR1, for example. Now, you might remember that restriction endonucleases cut DNA at palindromes, and so some people in the population might have a particular DNA molecule, particular RFLP allele that might contain 3 such palindromes, such that that gene will be cut at those 3 places by EcoR1, and from those 3 cuts we will get 2 small pieces of DNA. Whereas other people in the population might have a variation in that DNA sequence so that the middle palindrome has been eliminated. EcoR1 could still cut at the 2 outside palindromes, but not the one in the middle, so people with this DNA molecule would give us just 1 longer piece of DNA. We could measure the number and sizes of these DNA fragments produced by cutting with a restriction enzyme using southern blotting. So the different alleles will be distinguished by southern blotting. Panel B represents a type of DNA marker known as a VNTR. A VNTR, which stands for variable number of tandem repeats, also known as a mini satellite. In a VNTR, each one of these little boxes represents a base sequence that might be roughly 50 or so base pairs in length that is repeated several times in a row. When we have the same base sequence repeated several times in a row, we refer to that as a tandem repeat. And what this diagram is indicating is that for a particular VNTR gene, some people in the population might have that particular repeated sequence repeated 6 times in a row. Another person in the population might have that sequence repeated only 4 times in a row. Somebody else might have it repeated 10 times in a row. That's a naturally occurring, harmless polymorphism in the population. It is not necessarily related to any disease, and it does not mean that some of us are normal and some of us are mutant. It is just a naturally occurring polymorphism or way that we are different. Now, what is different between the 2 alleles for this gene is the length of that DNA molecule, and we can determine the lengths of those, the differences in the lengths of those 2 different alleles by southern blotting. Again, we will cut the DNA using restriction endonucleases and measure the lengths of the fragments produced by southern blotting. Panel C represents an STR or short tandem repeat, also often known as a microsatellite. Now, STRs are just like VNTRs, but they're smaller. In an STR, short tandem repeat, the repeated sequence is typically only 2 or 3 or 4 base pairs long. In this particular example, the repeated sequence is only 2 bases long, TG. And again, in the population, some of us might have, for a particular STR microsatellite gene, some of us might have that TG repeated 5 times in a row. Somebody else might have it repeated 3 times in a row. Somebody else 10, somebody else 15 times in a row. A naturally occurring harmless polymorphism in the population. STRs are smaller than VNTRs. We detect such STR differences by PCR, the polymerase chain reaction. Panel D represents an SNP, a single nucleotide polymorphism, or a SNP. In a SNP, there is just 1 single base pair difference in the gene comparing one person to another. These, again, are harmless polymorphisms in the population. We can detect these SNPs in the population either by DNA sequencing or by something known as ASO probes, allele-specific oligonucleotide probes. which will be discussed in Chapter 6 if you are not familiar with them already. So, these different DNA marker genes have been discovered as a result of the Human Genome Project. And now, as a result of that Human Genome Project, we have found at least 3 million of these different DNA marker genes scattered all over our chromosomes. And now, because of the Human Genome Project, we know exactly what these genes are and where they are located. And because we can detect them in the lab, we can use these marker genes sort of like mile markers out on the interstate highway. As you're driving down an interstate highway, you might look off to the right-hand side of the road and see a little green sign that will say mile 121, and then mile 122, and then mile 123. Those signs tell you where you are on the road. We can use these DNA marker genes like those mile markers, but on our chromosomes, so that as we move around our chromosomes studying various genes, by finding these DNA markers, we can determine where we are as we move around those chromosomes.